Good day everyone, especially to you sir. Today I am presenting to you the article entitled Growth and Yield of Oyster Mushroom of Pleurotus striatus on Different Substrates. The researchers are Ram Kailash P. Yadav and Chandra P. Pukril. I am Warren J. Claviti Bakad, your presenter. Introduction. Cultivation of oyster mushrooms or Pleurotus striatus has increased tremendously throughout the world because of their abilities to grow at a wide range of temperature and utilizing various agro-based residues. Pleurotus species are efficient lignin degraders, which can grow on different agricultural wastes with broad adaptability to varied agroclimatic conditions. Growing oyster mushrooms convert high percentage of the lignocellulosic substrate to protein bodies, increasing profitability. Of them, Pleurotus striatus demands few environmental controls, and their fruiting bodies are not often attacked by diseases and pests, and they can be cultivated in a simple and economic way. Mushrooms, with their flavor, texture, and nutritional value and high productivity per unit area, have been identified as an excellent food source to alleviate malnutrition in developing countries. Pleurotus striatus are a rich source of proteins, minerals, and vitamins. Apart from food value, its medicinal value for diabetics and in cancer therapy has been emphasized. Pleurotus species contain high potassium to sodium ratio, which makes mushrooms an ideal food for patients suffering from hypertension and heart diseases. Oyster mushrooms require less nitrogen and more carbon source. Thus, most organic matters containing cellulose, semicellulose, and lignin can be used mushroom substrate, that is, rice and wheat straw, cotton seed hulls, corn cob, sugar bagasse, sodas, waste paper, leaves, and so on. However, demanded amount of each nutritional source differs according to mushroom species and substrate use. The demand of mushroom has been mounting day by day due to population growth, market expansions, changing of consumer behavior, and developments. Rice straw is the principal substrate for oyster mushroom cultivation in Nepal. Hence, in this study, we attempt to identify the alternative substrate from various agricultural and forest residue and to assess the growth performance and yield as well as the nutrient content of Pleurotus striatus. Scope and limitation of the study. This study was conducted to investigate the growth and yield of oyster mushroom different substrates. The study was consist of 5 treatments and replicated 5 times. Treatment 1. Rice straw, which is control. Treatment 2. Rice straw plus wheat straw plus 10% of rice bran. Treatment 3. Rice straw plus pepper plus 10% of rice bran. Treatment 4. Sugarcane bagasse plus rice bran 10%. And treatment 5. Sodas plus 10% of rice bran. Materials and methods. Spawn or Pleurotus striatus was obtained from National Agricultural Research Council or NACR, called Maltar Lalitpur, Nepal for experiment. 5 treatments were prepared for experiment, that is, rice straw plus wheat straw, rice straw plus pepper, sugarcane bagasse, and sodas, all agricultural or plant-based residues. Rice straw was taken as control. Except control, other 4 treatments were supplemented with rice bran. Rice straw and wheat straw were obtained from agricultural field of local farmers in Kirtipur and chopped into small pieces 3 to 2 inches long. Newspaper was collected from the house and shredded into small pieces manually. Sodas was obtained from Patan Industrial Area, Lalitpur. After that, wheat straw and newspaper were separately combined with rice straw in the ratio of 1:1. Each treatment except rice straw was supplemented with 10% rice bran obtained from local rice mill. The mixture of substrate was soaked in tap of water and about 60% moisture was set to each substrate. 1 kilogram wheat substrate was filled in the polypropylene bag of 25 cm by 15 cm in size and autoclave at 121 degrees Celsius and 15 pounds pressure for an hour and allowed to cool overnight. After cooling, about 2.5% green spawn were inoculated on the surface of substrates and incubated in the dark at controlled temperature of 20 to 25 degrees Celsius. After colonization, the plastic bag was removed from the substrate and was placed in the growing humidity 70 to 80% and light intensity of 200 to 500. Mycelium growth, the total time taken from the colonization, primordial formation, and first harvest were recorded. Mycelial extension was vertically measured from 4 pieces at weekly interval for 3 weeks. The number and weight of fruit bodies were recorded. Total weight of all fruiting bodies harvested from 2 flushes was measured separately and calculated as total yield. The biological efficiency or BE was defined as the percentage ratio of the fresh weight of harvested mushroom over dry weight of substrate. The harvested mushrooms were oven dried and used for nutritional analysis. The ash, protein, fat, and fiber contents of mushroom samples were determined using standard methods. The total moisture was determined by drying the mushroom to a constant weight of at 105 degrees Celsius. Total carbohydrate and energy was calculated by using following equations. Carbohydrate equals 100 minus moisture minus total ash minus fiber minus protein minus fat. Energy equals protein times 4 plus carbohydrate times 4 plus fat times 9. Data were analyzed by using one-way ANOVA. 5 replicas were used for each treatment. Significant differences between treatments were determined by Torquay test at P less than 0.05. 5 different types of substrate were investigated to determine the growth and yield of Pleurotus austriatus. Weekly mycelia extension on different substrate is shown in Table 1. The fastest mycelia extension was observed in rice straw substrate, followed by mixture of rice straw plus wheat straw, sugarcane bagasse, mixture of rice straw plus pepper and sawdust, respectively. Mycelial growth is preliminary step that creates suitable internal condition for fruiting. Thus, outstanding growth of mycelium is a vital factor in mushroom cultivation. The Table 1 comparison of weekly mycelial growth of Pleurotus austriatus on different substrate. As you can see, the fastest mycelial growth is the control, which is the rice straw. Colonization of the substrate was completed in between 22.40 to 26 days of incubation. Similarly, primordial initiation on various substrate was also observed in between 26.40 to 31.6 days of incubation. The total day of the first harvest of mushroom took between 32 to 37 days, depending on the substrate used. The fastest colonization period, 22 to 0.40 days, primordial formation time, 26.40 days, and first harvest period, 32.40 days, were also recorded from rice straw. The colonization, primordial initiation, and harvest time are presented in Table 2. In our results, colonization and harvest time are not consistent with the results. Moreover, Xia et al. Of 2004 reported that primordial formation of Pleurotus austriatus occurs 27 to 34 days of inoculation, which is consistent with the result of this study. The size of protein bodies was higher in case of the rice straw, which is control, than in all other substrate. This is probably due to higher degradation of various constituents of the substrate rise through and consumption of high nutrients by Pleurotus striatus. Average mushroom yield of the two flashes, mushroom size and the biological efficiency are given in table 3. For each of the treatments, 2 flashes of mushroom were harvested. The yield was significantly highest obtained in the first flash of all treatments. The study revealed that the yield for rice through without supplementation was significantly highest yield among all treatments. The mushroom yield was not significantly different from the mixture of rice straw plus wheat straw and the mixture of rice straw and pepper. The highest yield, 381.85 grams, and biological efficiency of 95.46% of the mushroom was obtained from rice straw, followed by the rice straw plus wheat straw, rice straw plus pepper, and sugarcane bagasse. The lowest yield, 247.87 grams, biological efficiency of 61.96% was obtained from sodas. The lack of nitrogen may be one of the factors affecting the overall yield values in sodas. Sodas and sugarcane bagasse also contain high amount of lignin. Low degradation of lignocellulosic substances of sodas by Pleurotus striatus might be another factor affecting the overall low yield values from sugarcane bagasse and sodas. As fundamental food characteristics, ash, fiber, protein, fat, carbohydrate content, and energy value of analyzed of mushroom grown on different substrates are presented in the table 4. The study indicates that the fruit bodies are quite rich in protein, range between 22.89% to 25.97% on dry wet basis. Highest protein, fiber, carbohydrate, and energy were recorded from fruit body grown on rice straw. It has been reported that not only the protein content in fruit body but also the nature of protein depends on used substrate, according to Wang et al. Of 2001. Similarly, fat and fiber content range between 1.3% to 1.50% and 12 to 14% respectively. The highest fat was observed from the fruit body grown on sodas, whereas total ash was highest obtained from the mushroom grown on mixture of rice straw and pepper. The fat and fiber content obtained in the study are lower than that reported by Wing et al. Of 2001 as 2.50% to 2.82 and 5.97% to 6.42% respectively. The total carbohydrate content found in this study 30 to 24% to 42 to 26% is lower than the finding of Patil et al. Of 2010 as 50.50% to 55 to 33%. The total energy contribution of the sample range between 231.78 or 100 grams to 282.73 grams. Manji et al. Of 2001 reported that energy contribution of Pleurotus striatus was 30 cal or 100 grams, but also values of energy in calcium or gram were higher in comparison to his findings. In this experiment, different substrates have affected nutrient composition of Pleurotus striatus. This result may be probably due to biological, chemical differences and the CN ratio of the substrate, which is also indicated by several authors, Sangwan and Saini of 1995. Despite different Despite differences in the chemical composition of Pleurotus or striatus grown on different substrates, the overall nutritional potential of the mushrooms were relatively superior to several other research funding. Conclusion: mushroom cultivation is one of the efficient ways of which residues can be recycled. Pleurotus or striatus grown on different substrates are nutritious with high protein, fiber, and low fat. It may also offer economic incentives for agribusiness to examine these residues as valuable resources and development new enterprises to use them to produce nutritious mushroom products. Therefore, the mushroom cultivation may become one of the most profitable agribusiness that could produce food products from different substrates and help to dispose them in an environment-friendly manner. Almost all producers use rice straw for the production of Pleurotus striatus, which is also one of the best substrates in the study. Therefore, use of a variety of the substrates is essential. Although the amount of yield is lower than in rice straw, other substrates such as rice straw plus wheat straw, rice straw plus pepper, and sugarcane bagasse can also be used as alternative substrate with supplement in the cultivation of Pleurotus striatus. Thank you.